Proteins represent a class of molecules that have varied functions. Egg, muscle, antibodies, silk, fingernails, many hormones are partially or entirely proteins. Although functions of proteins are diverse, their structures are similar. All proteins are polymers of amino acids, that is, they consist of a chain of amino acids covalently bonded. One protein differs from another by the number and arrangement of the 20 different amino acids. Each amino acid consists of a central carbon bonded to an amine group, which is NH2, a carboxyl group, which is COOH, and a hydrogen atom. The fourth bond of the central carbon is shown with the letter R, shown here, which indicates an atom or group of atoms that varies from one kind of amino acid to another. So here are we can see in this diagram, here's the amine group, NH2, the carboxyl group, the hydrogen, and the R group. So the R groups differ in size, shape, and ability to form hydrogen bonds or ionic bonds. Because of the different R groups, each amino acid reacts with other molecules in a unique way. Notice we talked earlier about the ability of carbon to form double bonds in a carbonyl group. So this is a carbonyl group shown here. It's composed of a carbon atom double bonded with an oxygen atom. So it's CO. It's common to several classes of organic compounds as part of a larger functional group. In this case here, it's part of a carboxyl group. Okay, so the primary structure of protein is very important. It describes the order of the amino acids. There are 20 protein-forming amino acids that are assembled into polymers called peptides. The sequence of amino acids in a peptide chain is called the primary structure. So just as you have 26 letters in our alphabet to combine to form different words, these 20 amino acids can create an almost infinite number of combinations. Peptides can range in length from 2 to 2 million amino acids. And these are just rough values. An oligopeptide, oligo meaning few, would be 2 to 9 amino acids in length, roughly. A polypeptide might be 10 to 100 amino acids, and a protein is generally over 100 amino acids. Now these numbers are just kind of very approximate. There's no, textbooks like to come up with these numbers, but no one would really say that, these are hard and fast numbers of when something becomes a polypeptide. A polypeptide suddenly becomes a protein when it's 101 amino acids. That's sort of ridiculous. But these are generally sort of true. An example of a peptide would be antidiuretic hormone, or ADH, which is written as this sequence of 8 amino acids. Okay, so the primary structure was the identity of these different amino acids in a chain. The secondary structure is created primarily by hydrogen bonds between adjacent chains or loops. So you have hydrogen bonding that goes on that may lead to certain structure. Also, there are covalent bond angles between the amino acids that help to determine the secondary structure. So one form of secondary structure is an alpha helix, which you can see here. Another form is a beta strand, sometimes called a beta pleated sheet. So this forms a sheet-like structure. So these fibrous proteins can be beta strands or long chains of alpha helices. These proteins are not soluble in water. They're found in collagen, which is found in connective tissue in, say, your skin, and keratin, which is found in your hair and nails. And just as a side note, rhino horn, which is a big black market for, is composed almost entirely of keratin, which is the same material in your hair and nails. And it is a little bit unique. It is different than, say, a cow's horn. A cow's horn does have bone in the middle, and then an epidermal layer, and then the keratin, and then the skin and so forth. So rhino horn is somewhat unique in that sense, but really it's simply keratin, so it's nothing really that special about it. So it doesn't really make sense that people are hunting rhinos to extinction. Okay, the next level, so we had primary, secondary, then tertiary. Tertiary is even more involved three-dimensional shape. So for example, you might have a series of a number of strands that are alpha helices, and they may combine together via hydrogen bonding, ionic bonds, and so forth into an even larger structure. Like here we have something like collagen. Tertiary structure can also form what's called a globular protein. Globular proteins are roughly round. And often they're a mix of secondary structures. So for example, here in this globular protein, you can see the alpha helices, and you can see the beta pleated sheets, and then some other structures here. And so this leads to, and the interactions between these different parts via things like hydrogen bonding, ionic bonds, and the things we mentioned before, the angles, the covalent angles between adjacent amino acids of the angles of the covalent bonds would lead to these shapes here. You can also have disulfide bonds, and this happens when you have a cysteine amino acid and a cysteine amino acid. They can form this disulfide bond. And finally, at the highest level, you have what's called the quaternary structure. So amino acids combine with multiple subunits combined with non-covalent bonds. So a classic example of this would be the hemoglobin molecule, which actually has 4 subunits. So it has 4 globular proteins, so that you can see the different shades here of these structures, and they combine together to form one very large protein structure. And again, why do they combine together? Well, they're held together by hydrogen bonding, interactions among the R groups, and disulfide bonds. Okay, here's a question. Glycoproteins are made up of